Okay, welcome. Good morning, everyone. Welcome to our grand rounds. I will give a couple quick announcements and then turn it over to our speaker. So as always, if people have questions, feel free to type your questions into the chat function, and then I will relay those to our speaker, who also let us know that she's happy to answer questions as she goes. So if there's any questions about clarifications, please feel free to type those in. I also wanted people to know that next week our speaker will be Damien Fair, who is a developmental neuroscientist and neuroimaging researcher, formerly in Oregon, now at the University of Minnesota. And so it's a great pleasure today for me to introduce our speaker, Dr. Marina Picciotto. Dr. Picciotto is the Charles Murphy Professor of Psychiatry and Deputy Chair for Basic Science and a Professor of Neuroscience and Pharmacology at the Yale Child Study Center. Dr. Picciotto's lab studies the function of acetylcholine in relation to addiction, depression, learning, and appetite. Her work spans molecular genetics, biochemistry, cellular biology, electrophysiology, as well as animal and human studies, and highlights her philosophy that strong basic neuroscience can and should be clinically relevant. Dr. Picciotto has been, her work has been recognized across multiple spheres. She's a member of, or she was a member of the NIDA Scientific Council from 2011 to 2014. She has served as the treasurer for the Society of Neuroscience, president of the Society for Research on Nicotine and Tobacco. She's received multiple awards, including the Presidential Early Career Award for Scientists and Engineers, the Walensky Prize for Research on Substance Abuse, the Carnegie Prize in Mind and Brain Sciences. She was elected to the National Academy of Medicine in 2012, and she also currently serves as the editor-in-chief for the Journal of Neuroscience. So it's a real pleasure to welcome Dr. Picciotto, and with that, I will turn it over to you.
Thank you very much. It's a pleasure to be here this morning, and thank you for taking time to come online. I know how many Zooms we are all on, so it's really a pleasure to be able to talk to you about our work today. So I'm going to start by sharing my screen. I hope it is visible.
Yes, it is.
Wonderful. So as Dr. Posner mentioned, our work focuses on acetylcholine and what it does in various behaviors in various brain areas and how we translate that to understanding human psychiatric illness and human function. So to start, I just want to introduce the idea of neuromodulation. So acetylcholine neurons at the neuromuscular junction served for a long time as the model of presynaptic release opposed to a postsynaptic specialization where the motor neuron released acetylcholine, it diffused directly across to clustered nicotinic acetylcholine receptors, and there was a one-to-one relationship between the motor neuron firing and the muscle contracting. So when acetylcholine began to be studied in the brain, the thought was that the same thing would happen. It would be very much like a glutamatergic synapse. And it turns out that that's absolutely not the case. There are groups of cholinergic neurons both in the brainstem and in the basal forebrain that project extremely widely throughout the brain and innervate many structures, maybe all structures in the brain. And they are largely neuromodulatory. So what does neuromodulatory mean? I think it means different things to different people. For me, neuromodulation means that there is not semantic information content encoded in the acetylcholine signal. That is, when you have a sensory experience and glutamatergic neurons fire, that sensory experience is represented as faithfully in the brain. When acetylcholine neurons fire, they alter the likelihood of signals to be transmitted. They alter the length of time that signals can be transmitted, and they alter perhaps the way that different brain areas can fire together. And this is really reflected in a number of basic studies showing that acetylcholine signaling influences many, many aspects of brain function. So I alluded to this, that it contributes, acetylcholine signaling contributes to endogenous brain rhythms, and as well as synchrony of activity in limbic structures. There's a large literature on the ability of acetylcholine release and the necessity of acetylcholine release for attentional function. We know that acetylcholine can facilitate learning. In addition, inputs from the brainstem to the ventral tegmental area are essential for regulating the mesolimbic dopamine system and setting the threshold for reward-related behaviors. And as many of you may know, acetylcholine is essential for the various states of sleep and increases arousal during waking. And so all of these seem to be really good things. And yet, we also know that acetylcholine signaling increases anxiety and reactivity to stress. And I'll show you, that's what I will focus on today. So how can we resolve these important functions of acetylcholine in behaviors that are essential for survival for normal behavior, and the idea that it's also important for stress-related behaviors? Well, it's not stress that is bad, and I like this quote very much. It's not stress that kills us, it's our reaction to it. And in fact, stress is simply a challenge. It's a challenge to the brain and the body to alter behavior, to adapt to new circumstances. And this is something we do extremely well. I was amazed at the from day to day during the COVID challenge, we have adapted our behavior, and we've changed practices that we've had automatized for years in order to adapt to new circumstances. So stress simply changes our behavior, and good stress, eustress, is generally short, so we may be getting past that in the COVID challenge. It is beneficial. It enhances function, both physical and mental. You can think about this, for example, in strength training, where you stress your muscles. You may have some soreness, but over time you will become stronger. You'll build more muscle in order to adapt to those stresses. And we can think of stress as challenging work that allows us to actually reach to do better. But then when we talk about distress, now we think about the negative aspects of stress. It can lead to, if it's chronic, it disrupts homeostasis, it disrupts health. It leads to both anxiety and depression, and those are the states that I want to discuss today. So how do we model depression or anxiety in a rodent? Everyone on this Zoom has probably seen this figure of what a mouse looks like when it's depressed, when it's anxious. If I had a picture of a happy mouse, people would show the same photo. Of course, if we were really skilled at deciphering mouse facial expressions, we might actually be able to tell the difference between these. And there's some beautiful automated behavioral recording paradigms being carried out now that do suggest that there are big differences in facial expressions, even in mice. But one way is to actually start with a human, where we can ask questions about depression and anxiety. And so, in fact, when we started working on acetylcholine and responses to stress and the potential that it could contribute to depression or anxiety, there was actually a rich literature that we could mine in order to understand what increasing acetylcholine signaling, and particularly prolonging acetylcholine signaling, does in the human brain. And these were studies in the 1970s and 1980s. Janowski was one of the primary researchers who led this area of research. It started because there were epidemiological findings that pesticides that block, that are organophosphates that block the primary metabolic enzyme that breaks down acetylcholine in the brain and body. Exposure to those pesticides was associated with an increased incidence in depression in rural areas. And Janowski and his colleagues tested the hypothesis that it was blocking acetylcholinesterase that led to these symptoms. And so in a number of challenge studies, they administered physostigmine, a pharmacological antagonist of acetylcholinesterase. So it prolongs the amount of time that acetylcholine remains in the synapse, likely prolongs or increases the distance that acetylcholine can diffuse from its site of release. And what they found was that in normal human subjects, that is, people with no history of anxiety disorders or depression, administration of physostigmine could induce depressive symptomatology. This was also shown in people with a history of psychiatric illness and particularly of depression. And this led to a cholinergic hypothesis of anxiety and depression, where Janeski and his colleagues suggested that prolonged or too much acetylcholine signaling in the brain would set a threshold that would result in a greater likelihood of depressive symptoms, whereas increasing noradrenergic signaling beyond its homeostatic level would potentially lead to mania. And these hypotheses were explored and then quickly forgotten during the advent of the Prozac revolution. And that is when SSRIs, selective serotonin reuptake inhibitors, became the first line of medication for depression. The field shifted very strongly toward establishing how serotonin might be involved in depression-related behaviors and in therapeutics to combat depression. And the cholinergic hypothesis really fell by the wayside. And so I'm going to show you a number of studies that suggest that acetylcholine should not be discounted. And I think all of us are sophisticated now enough to say it's not simply one system that is dysregulated in any human psychiatric illness. It's not simply that we can regulate only one neurotransmitter and find that we have a therapeutic response. The reason we have a therapeutic response is that there's interaction between these modulatory systems, and so regulating one disrupts homeostasis in the brain and leads to adaptations in many, many different systems, one of which I would argue is the cholinergic system in the brain. So from the very early 90s, and this is one example, it was clear that exposing rodents to stress could induce acetylcholine release in many brain areas. And this is a study that was done with microdialysis, in which a microdialysis probe was put into different brain areas, the prefrontal cortex, the amygdala, the dorsal hippocampus. And what you could see is that acetylcholine levels were increased after the initiation of stress, remained increased for as long as that stressor remained on board, and actually likely outlasted that physical stressor, and then began to go back to baseline after that acute stress. What was interesting about this is that this appears to be independent of what we think of as the initial trigger for stress-related neurochemistry, and that is it was independent of the pituitary adrenocortical axis. So we think about pituitary function as initiating the stress response, but if you look at this response in adrenal-ectomized animals, so this is the hippocampal ACH release during restraint. If you adrenalectomize rats, you see exactly the same release of acetylcholine during restraint and then a rebound, in this case, after the restraint stops. So it suggests that there is a primary response to that stress without the intervention of the periphery in the cholinergic system. And so this led to a hypothesis that perhaps it's prolonging this acetylcholine signaling that is a potential model of maladaptive stress response in the rodent that we could use to interrogate the systems that might be contributing to these responses. And so we started out simply by trying to replicate Janowski's cholinesterase antagonist findings in mice. And so this is simply a pharmacology experiment where we injected physostigmine peripherally. We measured immobility in a model of antidepressant efficacy, the tail suspension test. Very simple. The animal is suspended gently from its tail. It tries to right itself. And if you give any of the antidepressant medications that are effective in humans, the animal continues to try to right itself for a longer time than the control animals. And you can see that the opposite is true when you give a cholinesterase antagonist. So this is a dose-response study, and as you increase the dose, you actually get less time struggling, more immobility time as you increase the dose. So increasing acetylcholine does the opposite of an antidepressant medication. And when we looked neurochemically at the cholinesterase activity in tissue removed from mice treated peripherally, what we could see is that at these higher doses, we see about half the activity of the cholinesterase. So likely the dwell time of acetylcholine was prolonged in the structures where we measured this. Okay, so we can at least replicate one aspect of Janowski's human findings. Can we be sure that this is a cholinergic response and that this isn't some other aspect of signaling? And so to understand the next experiment, you need to know that there are two broad classes of receptors for acetylcholine in the brain and body. There are the nicotinic acetylcholine receptors, which are ion channels activated by nicotine, hence the name, as well as acetylcholine at exactly the same site. And these receptors are generally excitatory when they're expressed on neurons and and acetylcholine signaling opens them. Those neurons are depolarized and fire. The second class of acetylcholine receptors are the muscarinic receptors. These are G protein coupled receptors, and they can either be coupled to excitatory G proteins or inhibitory G proteins. And on the cells in which these G protein coupled receptors are expressed, acetylcholine can either increase their firing, increase neurotransmitter release, or decrease their firing. And so what we asked simply was, could we block all of these receptors pharmacologically and reverse the effects of physostigmine in these mouse models? And the answer is yes. So here what you see is the same panel as before, and in our control animals, there's a certain degree of immobility in the tail suspension test. When we give physostigmine, we increase immobility, and then blocking the receptors for acetylcholine both reverse the effects of physostigmine. Sorry, that's here, either the nicotinic antagonist or the muscarinic antagonist or both together. And in fact, the level of behavior goes below baseline, and that is that there's an antidepressant-like effect in this very simple test of mouse immobility of simply blocking the receptors for acetylcholine on their own. And it suggests that if you decrease acetylcholine tone, not only do you reverse the effects of this broad antagonist of acetylcholinesterase, but you may in fact have antidepressant efficacy on its own. And that's consistent with some smaller studies of both scopolamine and mecamylamine in depressed human subjects, mostly as an add-on for the nicotinic antagonist to an SSRI. But thus far, these have not progressed to broad clinical use for a number of reasons. One is side effects, but also because the doses vary quite a bit in terms of their ability to block cholinergic signaling in the periphery and in the brain. So far, these are not a first-line direction for new therapeutics, but they are very useful in understanding potential mechanisms underlying aberrant acetylcholine signaling in depressed patients. So if this is going to be a model of behaviors that are relevant to depression and not simply messing with the brain of a mouse and changing acetylcholine signaling, we should be able to use antidepressant medications that do not intervene in the cholinergic system and see similar effects as we do when we block cholinergic receptors. So in this experiment, we added physostigmine, saw an increase in immobility, and reversed that with cholinergic antagonists. In this experiment, we saw the same increase in immobility with physostigmine, and now we were able to normalize that by adding fluoxetine, Prozac, as serotonin reuptake inhibitor that should not be intervening directly in the cholinergic system. And this suggests that whatever the serotonin reuptake inhibitor is doing to the brain, it is also able to reverse the effects of aberrant cholinergic signaling. All right, so these pharmacological data suggest that prolonging cholinergic signaling may provide a mouse model of depression-like behavior. Can we now start to use this to look at the neurobiology of where this cholinergic signaling is really intervening? And so what we did was to go back to the literature on where restraint stress increases acetylcholine signaling. And one of the most prominent areas was here in the dorsal hippocampus, where not only was there an increase during the stressor, there was this rebound increase even after the stressor was released. And we also did a survey of where cholinesterase activity was high in the brain and where it was increased after stress. And we really found that the dorsal hippocampus was a hotspot that was worth targeting more directly with molecular techniques. So what we did next was to, instead of intervening pharmacologically, which acts throughout the brain and body to prolong acetylcholine signaling, we used a small hairpin RNA targeted to the mouse version of acetylcholinesterase. We packaged this into an adeno-associated virus and infused it into the dorsal hippocampus. And what you can see probably vaguely on your screen is that there is a dark stain here in the hippocampus, and this is a scrambled control animal. And what you can see is that there's less dark stain in this animal that received the small hairpin RNA targeting the mouse version of acetylcholinesterase. We also made a humanized version of acetylcholinesterase, a cDNA, that could replace the mouse version so that we could be sure that whatever we did by knocking down the mouse version was selective for our intervention in the cholinergic system and not a broad effect of putting viruses in the hippocampus or of engaging the dicer machinery. And so we did rescue experiments as well, which I will show you later, with the human acetylcholinesterase gene. And what you can see is that when we measure hippocampal acetylcholinesterase, we have a significant decrease when we infuse the small hairpin RNA into the hippocampus. This is GFP that's also coupled in the construct that is packaged into the adeno-associated virus, and you can see this green color. When we do a single infusion of this particular AAV2 construct, it fills the dorsal hippocampus very nicely. So we have pretty broad knockdown, and I can't tell you very much about which subfields of the hippocampus are particularly targeted. And what you can see here is that when we do two models of anxiety-like behavior, they're both based on exploration of an open, novel environment. And one is the elevated plus maze, where one arm is closed, safe, dark walls, and the other arm is an open runway. What you can see is that knocking down the acetylcholinesterase actually increases anxiety-like behavior, very much like physostigmine did peripherally. Now we've only decreased acetylcholinesterase activity in the hippocampus. We've only prolonged acetylcholine signaling in the dorsal hippocampus. And now we have an increase in anxiety-like behavior in the elevated plus maze, as well as in the light-dark box, a similar exploration-based behavior where there's one dark chamber, one light chamber. And the more time the animal spends in the light chamber, the less anxious it is. The more time it spends in the dark chamber, the less anxious it is. And these are experimental paradigms that are sensitive, again, to human medications that are effective in humans to decrease anxiety. Similarly, in a number of models of antidepressant efficacy, we see the same trend or the same direction. And that is when we knock down acetylcholinesterase in the dorsal hippocampus, we see an increase in immobility in the tail suspension, which I mentioned to you before. In the forced swim test, again, a model of antidepressant efficacy in which the animal swims around in an inescapable environment. When it is given antidepressants, it explores that environment, tries to escape longer. And what we see is the opposite when we knock down acetylcholinesterase in the dorsal hippocampus. Going back to my description before of the rescue experiments, if we replace the mouse version of acetylcholinesterase with the human version, and that is we either include the human version at baseline or in combination with the mouse targeting small hairpin RNA, now we rescue this increase in immobility. And the same is true in the forced swim test. We also wanted to use a more ethologically relevant behavioral assay of stress response, and one that is sensitive to chronic administration of antidepressants as works in humans versus acute administration of antidepressants, which simply, I believe, captures initial neurobiological changes that later on would lead to antidepressant effects in humans. Here, the social defeat stress is a chronic stressor. It's a larger, aggressive mouse that's put in with a smaller, different conspecific mouse. And when the animal is put in with this larger, aggressive mouse over a period of between 4 and 10 days, what you can see is that after chronic social defeat stress, when we give the mouse the opportunity to interact with a non-aggressive conspecific mouse, they've learned to avoid that new mouse, and they decrease the amount of time that they spend interacting with a new mouse. And this is after 10 days of stress. After 4 days of stress, we actually do not see this effect. Instead, we see that the animal will interact with a new animal. And what we see is that when we introduce the scrambled, small hairpin RNA targeting acetylcholinesterase into the dorsal hippocampus, this short period of stress, 4 days, now resembles the long period of stress. So prolonging acetylcholine signaling in the dorsal hippocampus makes a suboptimal social defeated animal look like they've gotten 10 days of social defeat stress. And if we rescue the acetylcholinesterase signaling by putting in the humanized cDNA, now we rescue the behavior. So all of this taken together suggests that prolonging cholinergic signaling in hippocampus alone is sufficient to mimic the effect of systemic acetylcholinesterase blockade on these stress-related behaviors. And this is across all of the models that we've been able to look at so far of increased avoidance, increased immobility, or decreased social interaction. And so now the question is, do these changes in cholinergic signaling reflect something that we can see in patients? Can we see changes in acetylcholine levels, baseline levels extracellularly in depressed human subjects? And these are experiments that we did in collaboration with our colleagues a number of colleagues in the psychiatry department here at Yale, using a SPECT and now PET tracer that targets the same site on the nicotinic acetylcholine receptor that both nicotine and acetylcholine bind to. So this tracer was developed by a number of colleagues, both at Yale and at the NIH. It was validated in mice, in non-human primates, and then has been used successfully in both control human subjects, smokers, individuals with depression, and individuals with a number of other neurological and psychiatric illnesses to interrogate both the levels of acetylcholine in the brain as well as the number of receptors for that it targets. And so I'm going to tell you now about using it to interrogate acetylcholine levels. So if we accept the hypothesis that depression is associated in human subjects with increased acetylcholine tone, what might we expect to see with this PET or SPECT tracer when we're imaging these nicotinic acetylcholine receptors? So acetylcholine binds at the interface between subunits on these pentameric channels. And when there is normal levels or low levels of acetylcholine signaling, there are complementary interfaces that are available for the radioactive tracer to bind to. And so now when these subjects are imaged after a bolus plus constant infusion paradigm, we should see that there is receptor availability for tracer binding. However, if acetylcholine levels are higher in depressed patients, more of these interfaces should be occupied. And now when the tracer is introduced into these subjects, there should be fewer sites for it to bind. There should be lower availability for binding of the tracer. And that's exactly what we see. And that is that here's just one control subject. They have to be non-smokers. Here's one acutely depressed non-smoking subject. They have to be non-smokers because nicotine also binds to this same site and competes with the tracer. And what you can see is that patients who, in particular this patient, has lower levels of binding than this control subject. And I think I saw a question in the chat. So if there is a question, perhaps, Jonathan, you could read it for me?
Sure. I think the questions that have come up so far, I think probably we can hold off and answer at the end. They're not clarification questions at this point.
Okay, terrific. Then we'll come back to them. Okay, so this is summary data from one experiment, and I'm showing you one experiment with individuals who are either controls or who are acutely depressed at the time of the scan. And what you can see is that across the cortical areas and other deeper brain structures that were imaged, there is lower availability in acutely depressed subjects compared to controls. And this has been replicated now in 2 additional studies, one with individuals who are bipolar, but who are imaged during a depressive episode, and another in depressed patients. And what was seen in this particular study is that if a person was not depressed at the time of the scan, but had a history of major depression, major depressive disorder, their levels were intermediate across all of these brain areas. And so that actually is an important observation because it suggests either that the consequence of having been depressed is to have a prolonged acetylcholine signaling even during remission, or it may suggest that this is a potential biomarker that would suggest you might be more susceptible to developing major depressive disorder. So maybe environmental stressors or other precipitating events might make a person with elevated acetylcholine signaling more likely to become depressed. Now, we had to do a number of control experiments to assure ourselves that this was really a change in acetylcholine rather than a change in the receptors. So first, we did a post-mortem study taking cortical tissue from individuals who were either controls, who died in general from suicide, so who had a major depressive disorder diagnosis but died from causes that may or may not have been related to their disorder. We also took a group of subjects with schizophrenia because we wanted to make sure this was not just a general consequence of a brain disorder that was not necessarily associated with depression. And in this binding study, what we could do is completely wash out endogenous acetylcholine because we could, these were slices of human brain on a slide. And then we took the same tracer that was used in vivo, bound it to these slices. And what you can see is that there is no difference in the number of binding sites for this tracer once you wash out acetylcholine from the slice. Similarly, we wanted to ask if we challenged human subjects with physostigmine, as we saw in the Janowski experiments that led to increases in depressive symptoms, would we be able to also measure this kind of the same decrease in availability for binding? And this was a study with 5 patients within subject. And what you can see is that in each subject, before physostigmine, there was more availability, and after physostigmine, there was less availability. So it suggests that the challenges that led to prolonged acetylcholine signaling do displace this tracer even in normal human controls. So taken together, I think that this suggests that the mouse model of prolonging acetylcholine signaling is relevant to human depressed subjects. And now the question is, can we use this mouse model to identify the sites and receptors of cholinergic signaling that's important for these behaviors? So the hippocampus is innervated by these cholinergic neurons in the medial septum that are part of the basal forebrain complex. The basal forebrain complex and the medial septum itself also sends cholinergic projections to many other brain areas, in particular the cortex. And in general, these cortical projections of the medial septum are thought to be important for cognition, for attention, for things that we might not think about as important for or being involved in stress-related behaviors. And so what we did to start was to introduce a chemogenetic construct into the medial septum and ask whether we could increase activity of those neurons and see changes in these stress-related behaviors, as we did with the pharmacological challenge or with the knockdown study. And what we saw, right, so we're trying to look at this arm because that's where we introduced the small hairpin RNAs. And just to describe the approach a little bit more, the construct that we used was a Cre-dependent DREAD that is inactive unless it is expressed in neurons that express Cre recombinase, I'm sure all of you have seen these approaches before. We used an excitatory version in which the dread was coupled to GQ, and then we introduced it into the medial septum of mice that were positive for Cre recombinase only in neurons that express choline acetyltransferase, the synthetic enzyme for acetylcholine. And so in those cells, once the construct is expressed, we can peripherally inject CNO, which is an agonist of these chemogenetic constructs. And in those animals, we should increase acetylcholine release from the neurons expressing the dread. And what we saw in the first experiment was incredibly disappointing, which was absolutely nothing. So when we introduced this GQ or an inhibitory construct, a GI dread, into the medial septum, in these assays that I've already shown you, the light dark box, the forced swim test, and tail suspension test, we saw absolutely no change in behavior. So then we checked whether perhaps at least we could reverse the effects of physostigmine by inhibiting the medial septal inputs to the hippocampus. So if physostigmine is exerting its effects through the hippocampus, maybe decreasing the activity of the neurons that are releasing acetylcholine in the hippocampus would counteract its effects. There would be less acetylcholine in the synapse, and therefore prolonging the decreasing its breakdown would be reversed. And so what you can see is that with physostigmine, we see this level of immobility, and when we block the inputs, to the cholinergic inputs to the hippocampus, we can reverse those effects of physostigmine across these tests. So that was a small balm in this first experiment. But what we thought was perhaps what we're seeing, in fact, is the consequence of the broad level or the broad innervation of brain areas that might be involved in both adaptive responses to stress, as well as, in our case, we believe the dorsal hippocampus, which would be involved in maladaptive responses to stress. So when we infused the dread into the basal forebrain complex, we were likely activating all of these pathways. So then we wanted to take an approach that really pulled the hippocampus out. And one way to do that was, in fact, to introduce the chemogenetic construct directly into the hippocampus, into the same region where we saw the effects of blocking the esterase activity, and then using a virus that allowed it to be taken up by cholinergic terminals, again in a chat-free mouse, then we would only isolate the cholinergic inputs to the dorsal hippocampus. And when we did that, we saw quite profound changes in behavior that were very consistent with what we had seen with both pharmacology and small hairpin RNA experiments, both in the models of anxiolytic efficacy here in the light dark box, exciting the cholinergic inputs specifically to the dorsal hippocampus, decreased exploration time, so increased anxiety-like behavior, increased immobility in both the forced swim test and the tail suspension test, and decreased social interaction after subchronic social defeat. These are behaviors that looked almost exactly like the effects of knocking down the cholinesterase in dorsal hippocampus. And you can see that even though we injected the construct into the hippocampus, we are seeing significant expression in the cell bodies of the medial septum. So good. If we selectively activate this pathway, we can increase stress-induced behaviors. We also wanted to use a complementary approach. In this case, we used channel rhodopsin, a light-sensitive channel to selectively, that was infused into the medial septum. And again, the construct was put into mice. And in this case, what we did was to shine light on the terminals of those neurons in the hippocampus, and thereby increase the release of neurotransmitter only in the hippocampus after infusion of the channel rhodopsin into the basal forebrain complex. In this case, we're isolating that arm of the pathway with light rather than with a chemogenetic or a retrograde approach. And what we saw was the same direction, and that is in the tail suspension test in this case, what we saw was more immobility, just as we did with the chemogenetic approach. And so this is done in wild-type mice, so we were stimulating terminals that were both cholinergic and likely non-cholinergic, but once again, activating this pathway was sufficient to increase these stress-related behaviors. So we wanted to make sure that these behaviors were actually acetylcholine mediated. When you use the DREAD approach, when you use an optogenetic approach, you are stimulating the release of the neuropeptides that those neurons engage, of the circuit in which those neurons are embedded, and it's possible to change their activity and to have effects that are independent of acetylcholine. And so what we did was to do the same experiment, but to infuse an antagonist, in this case mecamylamine, the nicotinic antagonist, locally into the hippocampus after stimulating with the GQ DREAD. And so what you can see is that here in the light dark box, the tail suspension test, the forced swim test, we can reverse any effects of stimulating the medial septum inputs to the hippocampus by locally infusing a cholinergic antagonist. And this suggests that it is acetylcholine release that's initiating whatever processes in the hippocampus lead to these stress-related behaviors. Finally, I want to show you one more experiment where we're again asking, is it the cholinergic signaling in the hippocampus that's contributing to these behaviors? In this case, what we're doing is to infuse, again, a small hairpin RNA, this time targeted against one subtype, the most calcium permeable subtype of the nicotinic acetylcholine receptor family. We put that into the hippocampus. You can then challenge with physostigmine peripherally and ask if in the present or in the once the target in the hippocampus for these receptors is decreased, do we still see the effect of the pharmacological agent peripherally? And the answer is that in fact we can at least somewhat block the effects of physostigmine by knocking down even one of the nicotinic acetylcholine receptors in the hippocampus. And so here we see the effects of physostigmine in a control where the scrambled shRNA is infused into the hippocampus. And when we knock down alpha-7, we completely ablate the ability of physostigmine to increase immobility in the tail suspension, and we greatly decrease it in the forced swim test. And this is consistent with some of the peripheral pharmacology that I showed you early on when we knocked down acetylcholinesterase and then blocked its effects with antagonists of nicotinic or muscarinic receptors. So now I'm just going to tease data that I don't have to show for you because it is still ongoing, but what we've been able to do now is for a long time we could image the effects of the cholinergic antagonist, the acetylcholinesterase blockade by looking at measures of its activity. We could look at the consequences of acetylcholine signaling on cell firing with something like CFOS. But looking at acetylcholine levels in real time was something we really couldn't do with any precision in the rodent brain. And now Yulong Li's lab has developed a fluorescent acetylcholine sensor that can be expressed in the rodent brain. We've expressed it in the hippocampus to provide data that's complementary to the pharmacological and molecular data that I've shown you here. We've also expressed it in many other brain areas. It's a fantastic sensor. If any of you are interested in imaging acetylcholine activity, I'm happy to give you the information on how to get this sensor. But what we can do is to put this sensor in vivo, place a fiber over the area that we're interested in. In this case, I'm showing you data from the basolateral amygdala, and we can measure in real time the degree of fluorescence of this sensor, which is proportional to the amount of acetylcholine that is released. And so here, what I'm showing you is the consequence of an acute foot shock in this case, acutely on day one, and then after a fear conditioning paradigm, the response to that shock is, and the tone that predicts it is increased in the basolateral amygdala. And we can do this now with a number of behaviors and are currently doing this in these same paradigms to ask under what conditions is acetylcholine elevated, for how long is it elevated, where in the hippocampus is it elevated. Can we use that to start to build a circuit model and do some computational modeling for what it is that prolonging acetylcholine signaling in the hippocampus does to the network that might lead to these stress-induced behaviors. So that's where I'm going to finish. I've been talking to you about our work in the hippocampus. I showed you data on alpha-7 nicotinic receptors in the hippocampus that might be involved in these effects. I've shown you that the prefrontal cortex is, well, I haven't shown you directly, but we have other data, and I showed you some indirect data. The prefrontal cortex cholinergic signaling may be protective, in fact, against the behaviors that are induced by prolonging signaling in deeper brain structures. We've looked extensively in the basolateral amygdala, where we think that the basal tone of the structure is set by acetylcholine signaling. And I think overall what we would like to do is to get a coordinated, an overall model of how acetylcholine signaling from the basal forebrain complex coordinates the activity of these brain areas to result in both adaptive and maladaptive behavioral responses to stress. So just to summarize what I've told you, depressed human subjects appear to have elevated acetylcholine signaling throughout the brain. This doesn't appear to be directly due to one pathway or another pathway because it is so general. And I would be very interested to know if that is the result of changes in either the likelihood of release of acetylcholine, the degree of acetylcholinesterase signaling downstream, or synthesis or some other broad change in the cholinergic system. We know that acetylcholine signaling is critical for encoding negative information about the environment. That's been shown in other studies, and I think these studies show that when acetylcholine signaling is prolonged, negative information about the environment, stressful information, is acted on behaviorally more potently. And I've shown you with the chemogenetic and the optogenetic studies that stress-induced release of acetylcholine in the hippocampus can lead to these maladaptive stress-induced behaviors. And it leads us to our hypothesis that perhaps too much acetylcholine may contribute to the negative bias in encoding that is often seen as a core behavioral outcome in depressed human subjects. And why do I say particularly negative encoding? I say this because I've shown you a lot of data about the idea by the field that acetylcholine signaling is really important for cognitive function, for attentional function. And here I've shown you all of these stress-related behaviors where we don't really think about cognition as a core aspect of these behaviors. But we do know that a negative bias in encoding, taking the same information and encoding more of the negative aspects of that environmental stimulus than the positive aspects, can be something that leads to depression. And that's led to a model that we've developed where we think that acetylcholine signaling is absolutely essential for adaptive coping to the environment. We know that memory storage, reward processing, theta waves all depend on intact cholinergic signaling. And then as you increase the attentional, challenges to the system, perhaps there's surprising stimuli, perhaps there's stressful stimuli. Now you increase attention to those stimuli. You increase signal to noise, so you rule out, you screen out irrelevant information. And we've also shown that you maybe increase, at least acutely, stress reactivity in an adaptive way. And then as that signaling is prolonged, now we come to maladaptive attention to negative stimuli over rewarding stimuli. Probably also, and there's some data for this in the literature, impaired memory encoding. That is, that now we are not able to remember aspects of spatial navigation that are relevant to finding rewards and so on. And then maladaptive stress reactivity. And that all converges to change the encoding of threats. So I'm going to stop there and thank the people in the lab who did this work. I want to particularly highlight Jan Mineur, who is a longtime member of my laboratory. He's a research scientist in the group, and he's been absolutely essential in pioneering the studies on acetylcholinesterase signaling and in developing the ways that we can manipulate cholinergic signaling in the hippocampus that are relevant to stress-related behaviors. I want to thank our collaborators, particularly Zubin Bagogar, who was the first to use that acetylcholine signaling tracer in depressed human subjects and who really pioneered that work. Ralph de Leon has helped us throughout the years in developing these viral vector constructs. The late Ron Duman, who I miss very much, who passed away in February of this year, has been a longtime collaborator in all our studies of stress and depression-like behaviors. I Irina Esterlis did the studies on physostigmine challenge in human with the tracer, and Sherry McKee has done a number of lab studies with us. I'd like to thank our funders, both NIDA and NIMH, as well as the Brain and Behavior Research Foundation. You can follow me on Twitter for both updates on science as well as gifs of cats. And I want to thank you for your attention, and I'd love to take your questions now.
Well, thank you so much. That was really such an elegant series of studies, really incredibly interesting. Your work is so interesting in and of itself, but it's also just such a useful corrective against what I think is sometimes a myopic focus on monoamines within psychiatry. So thank you so much for that. I guess I will get us started with one question. Then I will turn it over to our chair, Dr. Lieberman, for some comments, and then open it up to the panel. So my question is, so you did a really nice job showing for us the relationship between stress and cholinergic signaling. And we usually think of early life stress as having sort of an outsized impact, a greater pernicious effect than adulthood stress potentially. And so I guess I'm wondering to get your thoughts on whether there are potentially developmental periods when the brain is more sensitive to cholinergic signaling.
Absolutely. And so we haven't done that extensively because our early life experiments have largely been confined to pharmacological interventions or molecular interventions rather than the early life stress that would be most relevant. But what we found is that there's a critical period for cholinergic signaling, which in the mouse is the first 2 weeks postnatal, probably corresponds to the third trimester in human development, where if we give nicotine, we see an enormous potentiation that is, as far as we can tell, long-lasting throughout life. We haven't seen it go away. That results in increased avoidance of foot shock. So it's a very simple assay. It's the passive avoidance test. And in animals that receive nicotine, so increased cholinergic signaling, during that critical period, we see a persistent change in the cortical circuits that process sensory information. And we've been able to dissect those circuits. We've shown that what happens is that during that critical period, we induce ASH2L and MEF2C signaling. These are a complex of that are considered to be an epigenetic marker where they increase acetylation of particular histones that we've then shown are associated with the promoters of genes that increase synaptic development and signaling through glutamatergic signaling. So what we believe happens, and what we've shown in these studies only of passive avoidance, is that too much cholinergic signaling results in opening up of the chromatin of all of these sensory links, and that persists into adulthood is not reversible by changing cholinergic signaling later. We've instantiated a program that is now independent of acetylcholine, and I think that's probably relevant to early life stress, but we have not directly interrogated early life stressors.
It's so interesting, and this is somewhat of a sidetrack, but one quick note: it's just in the human studies, it's been so difficult to tease apart the effects of prenatal smoking from shared genetics. So it's really interesting to hear about your work. But I wanted to turn it over to Dr. Lieberman for any questions or comments.
Marina, thank you for a really interesting presentation, and our condolences on Ron, who was really a towering figure in the field and at Yale, and it's congratulations on succeeding him. Big shoes to fill, but looks like you're certainly up to the task. So by virtue of my age, I can recall a little bit of a Back to the Future sort of theme in this, because when Janowski did that original work, and also coupled with the observation that anticholinergics can induce manic-like behaviors that seem to introduce a third neurotransmitter system into the mood disorders pathological search, along with the serotonin and norepinephrine. So what we've seen here, and also in relation to some new drug development in schizophrenia, is some non-paradigmatic targets being exploited for potential antidepressant or mood disorder therapeutic effects. Question that's arisen in the area of schizophrenia, for example, where a Karuna compound, which is an M1/M4 partial agonist, and then a Synovian compound, which is a TAR1 agonist, have shown efficacy, is whether they're acting through an independent, unique pathway or mechanism, or whether they're modulating the paradigmatic systems that have been traditionally used for the purposes of treatment development. Do you have a sense of that yet?
Yeah, I mean, I think the answer is that they are acting in concert with the noradrenergic and the serotonergic system, but the serotonergic and noradrenergic systems are also acting in concert with the cholinergic system. Let me first say that there are some very nice chemistry studies showing that many, and certainly the tricyclics are clear, but all of the antidepressant medications that are selective for norepinephrine and serotonin signaling actually have some affinity for nicotinic receptors as well. So there's likely, even at the level of interaction at the receptor level, an effect on anticholinergic. There's an anticholinergic consequence of many of the medications that are used for depression that are targeting the monoamines. What we've done is to try to ask, to what extent do we require intact monoamine signaling to get these effects of acetylcholine? And the answer is we do. So in the hippocampus, we've looked at 5-HT1A signaling, and if we knock down 5-HT1A, we now see synergistic effects with knocking down nicotinic receptors, for example. So subthreshold effects of either 5-HT1 agents or of the knockdown are exacerbated by cholinergic manipulations. We've also looked at interactions with noradrenergic signaling, and those studies I haven't talked about today at all. Those are all done in our hands so far in the basolateral amygdala, and we see the same thing. You absolutely require intact norepinephrine signaling in order to see any antidepressant-like effects of blocking cholinergic signaling. So I think that your point is very well taken. There are interactions between these, the cholinergic manipulations that we do and the normally targeted other neuromodulatory systems, the monoamine systems. And really what it suggests is that these neuromodulatory systems are overlapping in their actions. They're not simply redundant. You can't replace norepinephrine with serotonin with acetylcholine. The interaction between them is what allows us to provide an adaptive response to the environment rather than either no response or a hyperactive response.
Well, I don't know if Steve Roos or Tim Walsh are on, but the late Sandy Glassman published studies which showed that when smokers sort of try to withdraw, there's a increased frequency of depression as a consequence. So that's some ancillary evidence that seems to support your work.
Absolutely. I found Sandy Glassman's work when I first started working on nicotinic receptors in the early 90s and contacted him. We had some very good discussions on that. Let me actually answer some of the excellent questions that I see in the chat. And the first is about the fact that acetylcholinesterase inhibitors are used to treat Alzheimer's disease, and they don't appear to induce increased stress or anxiety, and that it's not consistent. So there are 2 points there. One is that in Alzheimer's disease, the tone is so low that we are actually on that spectrum of adaptive or really good, adaptive, maladaptive. We're starting here with Alzheimer's disease, and likely the cholinesterase inhibitors are not getting us very far past here. So I think that's one of the reasons that we don't see increased anxiety and depression phenotypes in patients with Alzheimer's disease treated with these cholinesterase antagonists. And then the other problem, and this is one reason that they're probably not as effective as we'd like them to be, is that there's also very rapid adaptation in the enzyme to pharmacological treatment. So when we see effects of physostigmine on the system, there's an increased transcription and translation of the acetylcholinesterase within hours, and it's maintained. So we blunt the effect of blockers of cholinesterase very quickly as a result of homeostasis. We don't see that when we use the small hairpin RNAs, and that's likely because we've really blocked the same mechanisms that lead to tolerance and adaptation when we bind the small hairpin RNAs to its target. So those effects last as long as we've looked. But interestingly, and I think that's worth exploring in some ways. So I just wanted to start addressing that question here. And then here's another excellent question around the pharmacology in humans, that muscarinic anticholinergic drugs are used to treat extrapyramidal side effects and were used to treat Parkinson's disease. There's no clinical evidence that anticholinergics reduced anxiety or depression in these patients. I'm actually not sure that that's true. I don't know how carefully that was looked at. Wayne Drevitz has done a number of smaller clinical studies using anticholinergic muscarinic drugs in a number of patient populations, particularly those with major depressive disorder. He's consistently seen antidepressant effects in those human subjects, and the biggest issue there was tolerance, that the side effects of the muscarinic antagonists are not really well tolerated by patients, and when you have other perhaps more well tolerated medications, that they're preferable. So I don't agree that there's not evidence for the muscarinic blockade leading to decreases in these depressive symptomatologies.
If you want, another question that came up from a few people is about the regional specificity. So in the human work that you showed, the effects seem to be quite broad, and then in your animal studies, you're focused on the dorsal hippocampus.
Right. So in the human studies and also in the mouse pharmacological studies, we're increasing we're decreasing acetylcholinesterase. So we're prolonging acetylcholine signaling everywhere, including cortex, where we think it might be adaptive. And yet we see these increases in stress-related behaviors, both in the humans and in the mice. In the DREAD study, we were only targeting medial septum. So I think there may be something particularly protective about the medial septum inputs to cortex, so that if we target those, both the hippocampus and the cortical only, that possibly that's why we don't see the effects of the chemogenetic manipulations. When we engage brainstem structures, the amygdala, the periphery, the autonomic nervous system, particularly the sympathetic nervous system, all of those systems are likely coordinating the activity of the body, the brain, subcortical and cortical structures in order to have this outcome that we read out in humans as depression or depression-related symptoms, and in the animals as these stress-related responses. So I think what happened was that the medial septum just happens to be a place where we do have more representation of the cortex and perhaps of these adaptive responses, and it will require more experiments to tease that out, where we actually put the retrograde dread into cortical areas to see, do we actually have a full-on protective effect of the dreads in some cortical areas? And we have some hints of that with early knockdown studies, but we don't have anything to hang our hat on yet.
Another question that came up was, what is your current thinking on the role of acetylcholine receptor desensitization versus acute acetylcholine action in mediating depressive and anxious behaviors?
Yeah, I actually think that's at the center of it. I think that's exactly why we have, first of all, the reports of smokers that they smoke to control depressive symptoms, and yet when you give them a handheld device and you ask them to rate their anxiety or depression symptoms across the course of the day, it's greater when there's, as an aggregate, it's greater when they're smoking than 2 weeks after they quit. So I think what likely is happening is you have these so let me just give background to those of you who are not nicotinic biophysicists. Nicotinic receptors bind to nicotine. They open to that rapid increase of nicotine in the brain that comes from smoking cigarettes or vaping from effective devices. After that coordinated opening of those channels, they're now profoundly desensitized for a much longer time than they were open. So you get a peak a coordinated peak of activity that leads to the dopamine release, it leads to the reward, it leads to the association of smoking with cues that promote smoking. But then you have this downregulation of those receptors for a long period of time. So my hypothesis is that you actually do cause some degree of anxiety, but briefly, during that period where those receptors are opened coordinately, but a much longer period of time in which you've decreased acetylcholine signaling through its receptors. And in fact, if you look over the course of the day with the same tracer and ask about occupancy of nicotinic receptors, the only time in a smoker that you have much availability of these receptors is first thing in the morning after a night of abstinence. These receptors bind to nicotine, nicotine's hydrophobic, it stays in the brain. You get actually quite a bit of occupancy of the receptors and therefore quite a bit of desensitization of the system all day. And then it slowly recovers during the night. So I think that the desensitization is really critical in any perceived antidepressant or anti-anxiety effects of nicotine. And again, there's a complex relationship because there may be activation of the cortex and particularly of nicotinic receptors in prefrontal cortex that are contributing to that feeling of decreased symptomatology.
Okay, thank you. There's another question from Alex Dronovsky, who notes that you began your talk with a focus on neuromodulatory actions of acetylcholine, but then focused on nicotinic receptors. Why did you select alpha-7, and what do you think is happening with signaling through muscarinic receptors?
Great question. So we've got so many receptors and only so much time, so we had to choose some to start. We are a lab that historically has worked quite a bit on nicotinic receptors. That's where we got into the acetylcholine field. And so we began with alpha-7 partially because of its calcium permeability and with the idea that it might be particularly contributing to plasticity. We've done experiments with M1 in other brain areas, in the basolateral amygdala and the other nicotinic receptor subtypes that are expressed in basolateral amygdala. In hippocampus, so far we've focused only on alpha-7 because of its extremely high expression in hippocampus compared to the other subtypes and compared to other brain areas, and because of its involvement in plasticity. But I don't think it's confined to alpha-7. It happens to be the one we used. And if you remember back to the pharmacology, when we provide physostigmine, or when we knock down acetylcholinesterase in the hippocampus and we give either the nicotinic antagonist or the muscarinic antagonist, and these are broad, they really block all of the subtypes. Either one alone is enough. So dysregulating the signaling downstream of acetylcholine is enough to combat whatever it's doing to these stress-related behaviors. So it's not clear to me why we can block either muscarinic or nicotinic receptors and get the same outcome. And that's one of the things that we'd like to do. So the alpha-7 is just a down payment on what we'd like to do more specifically going forward. Particular cell types, particular aspects of the circuit, and how does this work?
Great. I think this is probably our last question, and it's from Weili Cheng, who comments that human studies have typically implicated the ventral hippocampus in most affective processing. Have you compared your findings with ventral versus dorsal hippocampus?
We're doing that right this instant. So this is a question that I get quite often. I mean, the emotional responses to stressors should be largely mediated by the ventral hippocampus. I think actually the literature doesn't fully bear that out. Both dorsal and ventral contribute to emotional responses to stress, certainly a bias toward ventral. And that's one of the reasons that we came up with our idea of the biased encoding. Perhaps some of the more cognitive functions of the dorsal hippocampus are really important for what we're seeing in these effects of cholinergic modulation. So the experiments that we're doing now are using the acetylcholine sensor in both dorsal and ventral hippocampus during these same tasks, as well as now intervening with local infusion of cholinergic antagonists into dorsal versus ventral hippocampus to ask if there is a role for only dorsal. I doubt it. And if there is a role for dorsal and ventral, which I think is more likely, what's the difference? What aspects of the behaviors are contributed to by the two structures? And I have to say, we can't rule out that any of the manipulations that we've done with viruses exclude the ventral hippocampus. As you saw in that GFP labeling, the virus really does diffuse quite broadly throughout the hippocampus. So it's quite possible that what I'm presenting as the dorsal hippocampus infusion is involving some cells, maybe many cells in the ventral hippocampus as well.
Okay. So I think we will wrap things up with one final comment, which is not a question, but just a note from Sally Sattel that says, just a compliment, beautiful presentation. I couldn't say it any better. Really, such a elegant body of work and set of experiments. So thank you so much for taking the time to share your work with us. We really, really appreciate it. And just wanted to remind everyone that we will return next week with a talk from Damien Fair. So thanks, everyone, and have a great week.
I really appreciate it. Bye-bye. Great questions. Really appreciate that.